As we continue our discussion of evolution, we are going to switch from our smaller scale evolution, microevolution, and macroevolution, origin of new species, and switch over to learning a little bit about the history of life on Earth. In chapter 22, and in this lecture, we are going to introduce the origin of our first cells, which is actually in chapter 4. But I've moved it here because in chapter 22, we talk about big, broad, really big, broad patterns of evolution on the scale of millions to billions of years. And so I like to put origin of life into this category. So in this chapter, we will talk about origin of life. We will talk about the characteristics of the first cells and the theories for the origin of those early cells. And then we'll talk about some of the characteristics of those very early organisms. And from here, we will move into our diversity lectures, covering all of the domains and all of the characteristics of each of the domains and their phylogenetic groups within those domains. But before we do that, we are going to cover these big, broad patterns. And to get ready for that, we need to know where we get this information for these big, broad patterns. And the answer is mostly in the fossil record, although with current molecular techniques, the ability to learn information from DNA and from proteins, we can put that together with our fossil record and learn a little bit more about our organisms that we find and our hypotheses for the evolutionary patterns that we see. But we're going to find most of our information from the fossil record, especially when molecular evidence, DNA, protein signatures, etc., are not present. So a fossil is just the remains of past life on Earth. So they've been preserved in some way. Fossils tend to form in sedimentary rocks. So sedimentary rocks are rock layers that pile on top of each other. And when they do this, they compress down into really tightly compact layers. And different layers of sediment might be layered on top. And you can see in this image here that there are several different layers, different types of rocks. You can tell by the color. If you're up close, you can tell by the density. You can tell by the chemical makeup that they are different types of rocks. Older rock is usually deeper, and we can therefore infer that that older rock is going to contain older organisms. So a fossil is formed when the hard parts of the organism are buried, and then a chemical process occurs that essentially mineralizes the hard parts, and those hard parts form an impression in a rock face, or the parts are actually essentially changed into rock. And then, of course, they're dug up later, and we can look at the physical characteristics of those bones. Sometimes our geological activity that we see, earthquakes, plate tectonics, etc., can cause our layers to shift and slip, slide, even flip a little bit. So paleontologists really have to have a strong understanding of the rock layers that they're working with, and basically be able to carefully interpret the relative layers, put the rocks in the right layers, put the fossils in the right layers, and therefore get a relative date for their fossil. Fossils can also be dated using radioisotopes, such as carbon-14. If you are interested in that, there's a section in the textbook on carbon dating using radioisotopes to date some of our older fossils. There are several things that affect what's in the fossil record. Our fossil record is not complete. We're never going to have a record of every living thing that has ever been on our planet. But there are several things that make something more or less likely to have fossilized. So the anatomy of the organism is going to be very important. Something that has lots of hard body parts, think shells, skeletons, things like that, are more likely to be preserved than soft tissues because those soft tissues are probably going to decay much more quickly than the hard parts. You can see a few other things here. I'm not going to talk about all of them, but the environment is super important. There are specific environments that are more likely to fossilize, for the bones to fossilize. And so there are regions that are historic areas where fossils occurred much more frequently, just based on the weather, the water content, all of that. And then, of course, time is a big factor. Even if something fossilized, one, we have to find it, and two, we have to hope that it still remains intact or remains intact enough that we can see what it actually was. So something that is living more recently is more likely to be closer to the surface. It's more likely to remain intact. And so something that is from much longer ago is more likely to have been subject to geological processes, chemical processes that deteriorate that fossil. In your study guide packet, you should have a handout that is the same as this. It might be organized just a little bit differently, but this is a timeline from your textbook, and it shows the major evolutionary events in the history of life on Earth. History of life is broken down into time categories. The four largest categories are called the eons, and the three oldest eons are these down at the bottom, and they mark the Precambrian era. This is a dividing line in history that is basically or roughly delineated by the types of organisms. So if you look right here at the Cambrian era, before that, most of our organisms were small organisms. Many of them were single-celled, and for the majority of the history of life on Earth, organisms were single-celled. And then we have this Cambrian era. This is often called the Cambrian explosion. This is where we start to see a really huge uptick in the number of fossils, the number of vertebrates, the number of animals. And prior to that, early life evolved much more slowly. And then we see around this transition time a drastic increase in the number of organisms. To be perfectly honest, I will not ask you the dates on any of these eons. I do want you to kind of know that Precambrian, we have mostly prokaryotes and single-celled eukaryotes, and then all of the organisms that you are familiar with basically came out of this Cambrian explosion. Along with your study guide, there is a list of dates that you're going to need to know. On your handout, it'll show the dates, but you have to find them. It'll show the names of the dates, but you have to find them. So make sure that you check in with your instructor and make sure that you know what those dates are and that you get the correct ones if you can't find them on your own in your textbook. So prior to life on Earth, Earth had to form. It is estimated that Earth formed between 4.5 and 4.6 billion years ago. For most of that time period, there were not animals. Most animals evolved within about 500 million years ago or so. Eukaryotes evolved about 1.8 billion years ago. So for half to 2/3 of the time that Earth has been present, we've had mostly prokaryotes. But before there were any sort of organisms, Earth had to form. And on early Earth, the conditions were very, very different from the ones we see today. Earth's surface was extremely hot. There was a lot of land masses moving around, a lot of volcanic activity. The atmosphere around the Earth was full of rocks and gases, things that were decaying, radioactively decaying. There were a lot of asteroid impacts. So basically, it was just sort of a hostile, dangerous place to be. Not that there was anything here, but there was also no oxygen in the atmosphere. So the atmosphere was completely different than we see today. We have a lot of oxygen in our atmosphere and a lot less of the other gases, but it is thought that the atmosphere prior to the increase in oxygen was mostly carbon dioxide. And if you're in a semester where you've already done the ecology unit, you know that when there's high carbon dioxide in the atmosphere, we get the greenhouse effect. So not only was there all kinds of volcanic activity and radioactive activity, and you'll see below intense radiation, lightning storms, all of that, there was also a lot of carbon dioxide, which is sort of a side effect of all of that volcanic activity. And so when there's a lot of carbon dioxide, we get hotter global temperatures. And spoiler alert, if you're in a semester where we haven't learned about that yet, it's coming. So these are the gases that we think were probably present based on rocks that date back way back to this time frame. There are not a ton of them, but there are some places where you can get super, super old rocks. And there was not a lot of water. It was so hot that all of the water had vaporized. And so there was not a lot of water initially, but there were frequent lightning storms because there was lots of energy, lots of radiation, lots of ions in the atmosphere. And so there was a lot of lightning. And there was a lot of UV radiation from the sun. As Earth began to slowly cool and that volcanic activity began to settle down, the lightning began to settle down a little bit, oceans began to form because water condensed, that water vapor condensed and formed large pools. So relatively uninhabitable place until it cooled enough and had enough water, maybe even a tiny, tiny bit of oxygen. We don't really know for sure when oxygen kind of started to appear, but eventually it cooled down enough that life began. Our earliest fossils are estimated to date between 4 and 3.5 billion years ago. Now, it is very possible that there were organisms before this time, and there probably were organisms before 4 billion years ago. But the earliest fossils we have date from around 3.5 billion years ago. And those fossils are believed to be prokaryotes, simple-celled organisms, and resemble modern cyanobacteria. So we'll learn in a little bit that biologists aren't 100% sure if heterotrophs or autotrophs were the first organisms, if cells were doing respiration or photosynthesis first, but the earliest fossils are thought to be photosynthesizers at least. So whether photosynthesizers were the actual energy mechanism for the earlier ones, we don't really know, but we at least think that photosynthesis or the early beginnings of photosynthesis began in these fossil organisms that we see. So where did these cells come from? Well, there are several different theories. Our textbook talks about a couple of different theories for the origin of cells and where the biological molecules came from, where in or on Earth, what environments on Earth cells actually evolved. We're going to go through one of these hypotheses called the reducing atmosphere hypothesis. If you're interested in the other two that our textbook talks about, which is basically that our biological molecules came from an extraterrestrial source, meaning not on Earth, so from a meteor or from deep sea vents. You can read about that. It's super interesting, but for time's sake, we're only going to cover the reducing atmosphere hypothesis. I believe this is kind of the most commonly accepted hypothesis for origin of cells. So there are 4 stages to this hypothesis, and the first stage is that before you get cells, you have to have the molecules. And if you remember way back to the beginning of Biology 101, your class probably talked about organization and the levels of organization in biology. And at the lowest organizational level, we have atoms and molecules. And those atoms and molecules are then organized to make our biological molecules. And then our biological molecules are organized to make our cells. So before we can even get cells, we have to have the right molecules. So what we need are some biological molecules, like amino acids, like simple sugars, simple fats, simple nucleic acids. The reducing atmosphere hypothesis is based on the idea that oxygen begins to come together with carbon. Reducing means that an atom picks up electrons. Oxygen is really fond of electrons. It is very electronegative. And so oxygen likes to bond to other things. It likes to form covalent bonds. So reducing atmosphere hypothesis basically means that oxygen began to pull our biological molecules together, our 4 major atoms, carbon, hydrogen, nitrogen, and oxygen, together into these biological molecules. So to test the hypothesis that these organic molecules could come out of an inorganic source, 2 researchers, Miller and Urey, put together a really fabulous experiment in the 1950s. And it showed the potential results of a reducing atmosphere, an atmosphere where we have water, so we have oxygen, and that oxygen can come together with other molecules and produce our precursors to our biological molecules. So Miller and Urey simulated early conditions of life on Earth. Remember that there was no oxygen, or at least very, very little, if any at all, probably none at all. There was water vapor, hydrogen gas, methane, and ammonia floating around in the atmosphere around the planet. And they took these gases in what was thought to be a reasonable ratio for early conditions on Earth, and they built this closed system that is a series of essentially flasks and tubes, and they pumped the gases that they believed in the correct ratio to be present in early Earth. And then, because remember, there was a lot of electricity, a lot of heat, a lot of energy, they provided heat, boiling water to give us water vapor, and then also connected this apparatus to electrodes to simulate lightning. And so they basically were shocking these hot gases that are surrounded by water vapor. And then they let them drip through this tube on the bottom that is condensed, causes the gases to condense because it is cooled, and then collected down in a trap. So this is a closed system. So they know that the only things that are in there, what they put in there, there should be no biological molecules, there should be nothing living in there. And they collected the condensed gases into a flask at the bottom. And then they analyzed it to determine what, if anything, was in this solution. So they initially analyzed it and found that there were about 5 amino acids or amino acid precursors in this vapor that they were able to produce simply by heating up the water and the gases and providing an electrical current. So they showed that this could have happened. This sample was reanalyzed later in the 20 teens, I think, and they actually found in the sample that there were 22 or 23 amino acid signatures, including 10 that are current amino acids that are used to build our proteins. So they showed, Miller and Urey showed, that it was at least possible to build organic molecules from these inorganic gases. So stage 1 is forming those organic molecules. So Miller and Urey really gave us a hypothesis for how those organic molecules could have come about. Stage 2 is building our polymers. So remember that our large organic molecules are mostly polymers, meaning many units strung together. So for example, proteins are many units of amino acids, carbohydrates, many units of generally glucose, DNA and RNA, many units of nucleic acids. So those biological molecule precursors that would have potentially formed from these gas and water vapor would have to come together. We have to get the more complicated molecules. And so they need a place for this to happen. But building molecules doesn't just happen. You need some energy and you need a catalyst. You need something to cause this anabolic reaction to happen. You need a place for the molecules to be built. So in 1951, a man named John Bernal suggested that the catalyst for the building of these polymers was a special mud called montmorillonite, a special clay called montmorillonite. And he proposed that molecules within this clay and the mud that forms from this clay would have been a catalyst for the macromolecule formation. And a catalyst is a molecule that helps a reaction to go. In the biology world, these are usually enzymes, but remember, we don't really have proteins yet. In the chemistry world, a catalyst can be many things. It doesn't have to be a protein. And so he suggested that small pools essentially formed on the surface of this montmorillonite clay and catalyzed the formation of these organic polymers. So now that we have our organic polymers, we need somewhere for them to come together. And we need a cell-like structure to form a boundary to isolate those biological molecules and any activities that are eventually going to be our cellular activities in a single place. Because remember, our plasma membrane acts as a barrier to the outside. And that plasma membrane isolates all of the cellular activity. And so in order to have the activities that cells are going to perform, the metabolic pathways and processes, we need a boundary. We need to separate our cell from the outside environment. So when those polymers came together, they needed to first form a boundary. And so when we see basically a pre-living cell with a boundary, we call that a protobiont. So a protobiont is essentially a hypothetical precursor to our living cells, but these protobionts had to have a boundary. They had to have a boundary that isolates those precursor organic molecules, our new polymers, from the rest of the environment. Now, because all of our cells have a phospholipid bilayer, even if some of them are slightly different from one another, it is assumed that the protobiont would have had to have a version of the phospholipid bilayer. And that would have come about by phospholipid or phospholipid-like molecules forming polymers and then forming this lipid bilayer. Remember that the phospholipid bilayer is hydrophilic on the outside and on the inside layer, and hydrophobic tails on the inside. And that's what forces them to make these little pockets of cells. And then, of course, our polymers would need to be inside, and we need some special types of polymers. We need polymers inside that can carry information. We need polymers that can catalyze reactions, and we need polymers that can self-replicate. So this is saying that the protobiont would have had to have had the precursors to our nucleic acids and to our proteins in order to be able to do all of these things. And when you have all of these things, you have the protobiont. Stage 4 is having a coding molecule. So just like the process of natural selection in the mechanism of evolution, molecules can undergo something called chemical selection. And basically, the idea of chemical selection is this protobiont may have had some molecules that were starting to do metabolic type reactions, like forming enzymes to be catalysts and forming nucleic acids that could contain information. And when there was a molecule that worked particularly well inside a protobiont, that molecule, which now has the ability to self-replicate, could proliferate on its own. So this idea of chemical selection says that there are some molecules that do a really great job at being the precursors to living cell molecules. And so it is hypothesized that the very first biological macromolecule was probably RNA. And you're probably thinking, well, don't all cells have DNA? They do. All cells now have DNA as their genetic material. But the reason that RNA is thought to be the first biological macromolecule is that because it's a nucleic acid, it can store information. In our cells now, that's not fully its job, but it can and does store information. And then because it is a nucleic acid chain and works on the principle of complementary base pairing, its sequence allows for self-replication, just like the DNA molecule does when your cells replicate its DNA. But the other more important or interesting reason that we think that RNA was probably the first biological macromolecule is because of a type of molecule we find today called a ribozyme. A ribozyme is an RNA molecule that functions as an enzyme. So this is not a protein enzyme, this is an RNA enzyme. And because RNA has this ability to catalyze reactions, it could have potentially both stored information and acted as an enzyme. It could have done both jobs, whereas neither DNA nor proteins today can do both jobs at the same time. So this hypothesis that RNA was the first biological molecule is called the RNA world hypothesis. And this is the hypothesis that RNA came first and both coded the information and performed early metabolic activities. And then eventually that RNA was replaced by DNA and proteins as the main information and catalytic molecules. Modern DNA is far more stable than RNA, and so it would be beneficial for the cell to have a more stable molecule containing its genetic information.